लाइसीमिया, एरीजिया, ये सारे नॉन लैक्टोज फर्मेंटेड। तो हम कैसे पहचान करेंगे शिगेरा को? We will use a highly selective medium known as deoxycholate citrate agar, which is going to give you translucent qualities, or XLD, that is xylose lysine deoxycholate agar, which is going to give you red colored qualities without black center. Salmonella shigella agar, which is going to give you yellow colored qualities, and Hector enteric agar. Okay. So these are the other selective media onto which your shigella is going to grow and give different kind of colors. Okay, laboratory diagnosis, it is going to give you short, gram-negative bacilli, non-motile, non-capsulated and non-sporing. Biochemical reaction, since it belongs to Enterobacteriaceae family, it will be catase positive and oxidase negative. So catase positive except Shigella dysentery serotype A. So you need not remember the exceptions. Just remember ये family Enterobacteriaceae को belong करता है, so it will be catase positive, oxidase negative. Okay, and manner of fermentation. So do not ferment lactose and sucrose except Shigella soni, which is a late lactose fermenter of both. Okay, so it will be indole negative, urease citrate negative, and there will be K by A. So your slant will be alkaline, your bud will be acidic. Okay. The oxycarbonate test, so we when fermented, given your amino acid, lysine, arginine and ornithine are negative except Shigella soni, which decarboxylate your ornithine. Okay. So treatment, ciprofloxacin is the drug of choice, and oral dehydration solution, prevention, hand washing and stool decontamination. Okay. So are we clear with dysentery? Okay. So as we are going to study more organisms in microbiology, we are going to come across with more organisms which are going to cause UTI. So you need to make a note of this, because the question will be asked like, what are the 4 organisms which will be causing urinary tract infection? हमने तो सिर्फ एक ही पढ़ी, but जैसे जैसे हम माइक्रोबायोलॉजी पढ़ते जाएंगे, we are going to cover virology, we are going to cover mycology, we are covering bacteriology, right? So जैसे जैसे we are going to cover microbiology, you are going to get organisms which are causing diseases in a specific particular system. Okay? So the question is going to come like this: enumerate 4 organisms causing UTI and give the laboratory diagnosis of E. coli. Okay? So the most common organisms are laboratory diagnosis, but you are also supposed to enumerate the other causes of UTI. Okay? Enumerate the organisms causing dysentery. Okay? So it will be amoebic. Okay? Parasitic dysentery. It could be bacillary dysentery. तो आपको सारे causes लिखने हैं, but give me laboratory diagnosis of Shigella. Okay? This will be like this. So we have covered 2 systems per se: urinary tract infection and dysentery. Okay? Are we clear in this? Can you write the laboratory diagnosis? So you are supposed to write the gram stain, then the culture, then the biochemical reactions and EST if it has any antibiotic resistance. So here under 5 heads it should come your answers. Okay. Next we will go to Klebsiella. It is one of the organisms causing pneumonia. Klebsiella pneumonia. In the name itself it has pneumonia. Genera, it has Klebsiella, Enterobacter, Hackney and Serratia. Okay. Klebsiella is found in the commensals in human intestine and as in saprophytes in soil. It has 2 species, one is Klebsiella pneumoniae and Klebsiella oxidova. So this is one of the lactose fermenter. What it is going to give colony as pink colored colonies. But it also has a capsule, Klebsiella pneumoniae. So the pink colored colonies will be sticky or mucoid, very big. Okay. So this is non-motile and capsulated. Okay. Klebsiella pneumoniae, subspecies pneumoniae. It is most pathogenic. It causes severe lobar pneumonia, urinary tract infection, meningitis, septicemia, and pyogenic infections such as abscesses and wound infection. It colonizes the oropharynx of hospitalized patient. तो hospitalized patient में ये जाके colonize कर जाता है. So what it is going to give you? hospital acquired infection. Okay, जो patient में his immune system has gone down and the patient are admitted for more than 1 month, what it will go and do is colonize wherever the external devices are present. Okay, supposedly the patient cannot breathe himself, so what will he is put into ventilator. Ventilator के बहुत बड़ा ट्यूब अंदर जाता है. Okay, so this Klebsiella it will go and reside onto the outer surfaces or inner surfaces of that ventilator tube. Okay, फिर कितना भी हम antibiotic दे दें, it is not going to go to the patient. Why? Because it forms a biofilm over there, and the biofilms are known to not penetrate. The antibiotics will not penetrate in the organism and not kill the organism. Okay, so that's why it's a common cause of nosocomial infection. So most hospital strains are multi-drug resistant because of biofilm formation. Okay, so how are you going to diagnose pneumonia? What is the sample you are going to collect? Sputum. Okay, and will come the other samples like bronchial washings, bronchial brush biopsies, gastric lavage. Okay, and since it is a systemic infection, you are also supposed to collect blood. Okay, sample से क्या करेंगे पहले? Culture में डालेंगे so that it does not get contaminated. On the first day itself, it is put onto the culture, meconium or blood, and incubated. क्योंकि तभी की तभी वो ग्रोथ देगा क्या कल्चर कॉलोनीज में? नहीं, उसको टाइम लगता है वो न्यूट्रिएंट लेके, because we have studied the bacterial growth curve. So it takes some time to take up the nutrient and multiply. Okay, so we are given 24 hours incubation at 37 degrees Celsius in the incubator, time for the organism to grow. Okay, so first it is plated onto the media, and second we do a gram stain. Gram stain is, since it is a gram negative bacilli, how are we going to see? So you can see, since this is a capsulated organism, gram stain around the bacilli, it is clearing. Okay, and you can see G and B which are inside, gram negative bacilli which are inside. Okay, the capsule. You can see clearing around the bacilli. What are these? These are pus cells. Okay. So yes, this is how a gram stain picture looks like, that of a sputum. बहुत सारा pus cells होगा, epithelial cells होंगे and bacilli होंगे. Okay, am I clear? So the gram stain is going to show you short, plump, straight, capsulated, fragmented rods. Clear? Okay. After that, you can see this is a McConkey plate and very big colonies you can see. Okay. E. coli कैसे थे? छोटे colonies थे या फिर spreading colonies थे. These are very big. You can make out a single colony, and these are dripping. Okay, almost dripping from this thing. So this consistency gives you to any capsulated organism. No, this is going to give you mucoid-like consistency. Okay. So due to the capsule, it has mucoid colonies. So large dome-shaped mucoid due to capsule, sticky pink color lactose fermenting colonies. Okay, this should come in your answer. Now this is indole negative. One peculiar thing, it is citrate positive and urease positive. Okay, so citrate and urease positive. Triple sugar ion, it produces enormous gas. So आपका जो पूरा TSI है वो पूरा येलो होगा, न्यूट्रल रेड इंडिकेटर TSI में भी डलता है. So due to enormous acid production, everything will turn into yellow. Straight, sit straight, sit straight. If you are feeling sleepy, stand up, come on, stand up. Keep standing for 5 minutes. Okay, no leaning on your friend's head. This is a class. Okay, even if it is an honorary lecture, it is a lecture. I am not wasting my time or your time. Okay, keep standing for 5 minutes, and whenever you feel like you are not sleepy, you can sit down. Fine. So triple sugar ion, it is so much enormous gas production, it is going to lift. आपका छोटा सा ट्यूब रहेगा, the media will be lifted up, इतना gas production रहेगा. Okay, कभी कभी इतना test tube रहता है, the media is over here, इतना enormous gas production होता है. Okay, so due to enormous gas production, you can make out